and so on. So it will be a bit technical, but not too advanced. So we'll try to cover as much as possible. So if you have any questions during the presentation, you can interrupt me. Otherwise, we can discuss it at the end as well. Okay, so I'm in charge of the throughput screening facility here at King's. And so for us scientists, the start is always the biological question, right? And for us, screening is just another way to address this biological question, and conceptually a very interesting way to address biological questions. So there are, of course, other approaches. The classical approach, where you generate a hypothesis, you test, then you formulate a new hypothesis, and so on. This, of course, is very useful, but is always limited or influenced by the priori knowledge that you have on a particular subject. Then you have other types of approaches, like the multi-omics approaches, where what you're doing there is characterizing the system in terms of different biological species. You can do genomics, transcriptomics, proteomics, any kind of omics now, single cell as well. But really what we're looking at is at the different abundance of the different molecules that you are analyzing. And based on that, you try to infer, or we try to infer function. When we do a functional screening, we're actually screening for function. We're really perturbing the system in a systematic way, either with small molecules or with genetic or genomic screenings. And then what we are able to evaluate is the relevance of each of these perturbations to the phenotype or to the cellular process that we are studying. And more than that, we can, without any a priori knowledge, we can rank the molecules by their potency. So even if we could not anticipate that this molecule was important, or that this gene or this protein was important, by doing this unbiased screening, we can get to this molecule and knowing that this is the most potent factor. This, of course, has a great potential for novel discoveries and for really putting us in a different plan and discover new biology. So when we speak about screenings, we have two types of screenings in general. One is what we call pooled screening and arrayed screening. Okay, so we'll start with the pooled screening. Pooled screening is typically done when we have libraries of oligonucleotides that then are cloned into lentiviral vectors. And the idea here is that the library is produced as a pool, and cells are also transduced with this pooled library. So we will end up with cells that express different siRNAs or CRISPR guides, for example. And so each of the cells will be different. And then what we have to do is to apply some sort of selective pressure. This is typically proliferation or cell death or sorting, some kind of separation. And then the readout of this screening is based on sequencing. So basically what we're looking at is, in response to a specific stimulus, what are the cells that are going to be selected either by survival or decreased death. And so by that, we can then recover these sequences and understand that the genes that were modulated in this selected population have their own effect. So one strong limitation of these types of screenings, as we'll see in a minute, is that they are limited by the phenotype that we are analyzing, because we always need some kind of selective pressure, either through survival or we can also do physical separation of the cells by flow cytometry. But they are very interesting for some systems, and I'll come back to this just in a minute. Let me just introduce you array types of screenings, and then we discuss advantages and disadvantages of both. So in the array screening, what we do is that we have libraries of different reagents, so libraries of chemical compounds or some other molecules that will perturb function of the cells. These are separated in different wells, and so each population of cells in each well will receive a different treatment. And then what we analyze after is the consequence of this treatment to the phenotype of the cells. So we call this one perturbation, one well approach. And here, the advantage is that we'll have an homogeneous population responding to a single stimulus. So advantages and disadvantages of both of these approaches. So as you can imagine, the array screenings, and as you'll see in a minute, allows for very complex phenotypic readouts, for example, microscopy, as you'll see in a minute. Whereas, as I discussed before, the pool screenings are restricted to some sort of selection phenotype. In terms of the libraries, for the array screenings, we can get very good quality libraries. These are usually synthetic libraries that can be bought, and we can have flexibility in the coverage of the libraries. Whereas these libraries for pooled screenings, these typically need to be cloned into plasmid DNA that has to be prepped, and then we have to generate lentiviral particles typically for these types of screenings. In terms of cell target, because these molecules here in the rate screenings, if we're thinking of genomic screenings, we need cells that are able to be transfected. This is not an issue if it's a drug screening, but if it's a functional genomic screenings, we need cells that are transfectable. Whereas here, because we're using lentivirus, we can also transduce difficult to transfect cells, which is a great advantage, as well as the duration of the silencing or gene modulation, other types of gene modulation, where here is transient because it's a synthetic molecule, and here is more permanent because it's a vector. Both are labor intensive, different types of work. Here it's all the automation and so on, and here is the prepping of the virus and the plasmids. Different infrastructures are required. Here we require an infrastructure for high-throughput screening, here access to an NGS platform. So there's no one size fits all, so every screening can be, one particular question may be better addressed by one type of screening or the other. Now, the screenings that we do here are all of this type of array screenings. We have different types of libraries, as we'll see. And so everything that we do here in the lab is of these array types screening. And using this, this gives us a lot of flexibility in terms of the phenotype that we can test. So we can really look at multiple processes. And here I'm just listing some, looking at proliferation, cell cycle, gene expression, apoptosis, autophagy, and you name it. We have to design an assay for that, but this can be measured by this technology. So this is the kind of processes or cell pathways that we can analyze, and we can analyze also these through different methodologies. Okay, that can go from very simple assays run in a plate reader, for example, where we measure viability of the cells or expression of a given reporter or something like that. And then they can go to more and more complex readouts, for example, microscopy-based readouts, or even more morphological or phenotypic readouts of the cells, and I will now explore this a little bit more in detail. Again, there are advantages and disadvantages to all types of readouts. The simplest readouts that we call plate reader-based readouts, where we get a single read for the whole population of cells in the well. So this typically involves lysing the cells and then measuring. Typically, this is absorbance or luminescence or fluorescence to give, reported gene activity, survival, and so on. They are very simple. They are fast. They are easy to optimize, but the results are very binary, right? So it's just an average of the cell population. If we go in more detail towards what we call high content screening, so image-based readouts, here we still are quantitative. We can get much more information. So we get multiparametric information. We get subcellular resolution, so we can look at different compartments inside the cell, and we get single cell resolution as well. So we get information for every single cell. This is, of course, an advantage and allows us to do much more refined phenotypes, but comes with more complexity and typically higher cost, and also the analysis is more complex. Then there are other category of screenings in which we don't really have a primary readout. So for example, here we can screen for specific readouts, right? So morphology of the cells, proliferation, translocation of a particular transcription factor or molecule or whatever. Here the idea is different. That is, let's just profile the shape of the cells, the general shape of the cells as much as we can, and see how this changes between different treatments. And then we don't know what's happening to the cell. We know that it's changing, so we get the signature of each change with each treatment. And then we try to compare these different signatures to come to an effect and to compare it with known compounds, for example, to infer a mechanism of action. So I'll get back to this so that this becomes a bit more clear. The advantage of this is that this is a very generalized assay. So we're just staining the cells, whatever cells are, the same way, with the same dyes, and so on. We just image them, get all information that we can, and then we analyze the data later. So we image first and ask questions later, okay? And the disadvantage is, of course, that we don't have a primary readout. So we're looking at changes of cell states. For high content screening, so for the image-based screenings, this is straightforward. We go from the original image to a computer-processed image. We express multiparametric analysis from all these images. As I said, we get subcellular resolution, single-cell analysis. For each of the cells, we get numerical data. Okay, so we go from the images through to the numerical data through these image analysis softwares. And this is really for us the sweet spot for doing screenings, because we know what our primary phenotype is. We know exactly how to optimize the assay for that particular phenotype, and then we just go with a more sophisticated microscopy readouts rather than going for the cell profiling. But in some instances, this could also be useful. So the image-based cell profiling, so as I mentioned, so we measure first, we ask questions later. So what's the idea here? We have a population of cells stained with different dyes, standard dyes. We analyze the cells using the same image analysis software, and we get these morphological features and signatures relative to each treatment. So now we can compare treatments of cells without knowing what each treatment is doing. Right? So then we can do, 2D projections to analyze similarities between different treatments. We can do all sorts of things, or we can also query against reference molecules so that we understand that treatment X is similar to a known drug, and then from there we can get to the mechanism of action. This usually involves staining of the cells with different dyes, and the most used assay for this is called the cell painting assay that you may have heard already about. So this is an assay that has been developed at the Broad Institute, and it's basically, it uses 6 fluorescent dyes. So these are not antibodies, these are dyes. So this is very important because it's cost effective. And these dyes label 8 subcellular structures that you see there in 5 fluorescent channels, right? So we stain the cells with these 6 dyes, we image them in these 5 wavelengths, we extract as many features as we can. We're talking about 1,000 independent morphological measurements per cell. Okay, then we aggregate these to create those signatures that I was referring to in the previous slide. Okay, so this is now a very much streamlined and standardized assay for doing this kind of self-profiling.
Sorry, can I ask just
Sure.
because for one cell you have 1,500 morphological measurements. Is this to do with like subcellular distribution of the organelles and their relative positioning to one another and that kind of?
Yeah, you can go in different degrees of complexity, right? You can imagine that we cannot rationalize 1,500 parameters for a single cell, right? So what the computer is doing is everything that you said in terms of positioning and so on, also in terms of intensity of the relative dyes, also in terms of texture so the distribution of the signal, also radial, spatial distribution within the cell, closer to the nucleus, farther from the nucleus. So these are several layers of analysis that end up with this number of parameters. It happens that some of these parameters are not independent, right? So they are very highly correlated. And so later on, before doing the profiles, these highly correlated features have to be reduced so they don't create a bias in the data, right? But the idea is that we can extract a lot of morphological measurements per cell that will end up creating these different signature profiles, right? So if you want, you can do an analogy with what is a gene expression profile, where you have 8,000, 10,000 different measurements for one cell. Here we have 1,000 different measurements for one cell, but these are morphometric intensity-based measurements.
How specific are the dye binding to the subcellular structure?
How specific are?
Yeah.
What? Sorry.
How specific? Because this dye we binding to mitochondria, right?
Yeah.
We combination with SR. So how specific?
Yeah, I mean, so these dyes are, standard dyes. You see the names here, right? So these are WGA, mitotracker, concanavalin A, and so on. So they bind to these structures. If they bind differently in consequence of the different treatments, this is already a feature, a signature. So this will shift your staining, standard staining, towards a different staining. That will tell you maybe that the mitochondria have a different shape, have a different polarization, have a different component, okay? So we don't know how this changes with different treatments, but I mean, because the dyes are standard, they will react the same way every time with similar structures, with similar phenotypes. So it's agnostic in the sense that we don't know what we are measuring, but is super powerful. And so this is like the gold standard assay for these types of approaches. But there's been even further developments on this. For example, people looking at cancer cells have developed this technique that allows for staining with 40 different antibodies, the same cells. So now you can imagine a matrix where you have stainings from 40 different antibodies, single cell, again with multiple parameters extracted per cell. And they do this with standard antibodies with basically 20 cycles of IF washing, and bleaching, washing, and new AF, and so on. So of course, this is very sophisticated and very difficult to implement, but just to tell you that it's possible and this can be increased in terms of complexity. Okay, so shifting a little bit gears and speaking about the libraries. What are the libraries that are available? What kind of libraries can we screen? Again, here there's multiple choices. We can either use libraries of synthetic compounds, so small chemical compounds. It's also possible to screen with natural compounds. They have the issue that they are typically mixtures, so then you have to somehow deconvolute the composition of these extracts. When they are pure compounds, this is not a problem. We can do libraries of genomic libraries. This could be siRNA, microRNAs, CRISPR, cDNAs, and so on. We can do, for example, pathogen collection. So we're using the same cells and the infected cells with mutants of bacteria, for example. And here, even inside each of these types of libraries, for example, if you consider chemical libraries, there are different sets of libraries. So there are libraries which the objective is to be as diverse as possible, to explore the chemical space as much as possible. And there are more focused chemical libraries, for example, libraries of biologically active compounds, so compounds that we know already where they act, compounds that have been approved by the regulatory agencies, or, for example, more focused compound libraries, for example, kinase inhibitors or epigenetic modulators. So these types of libraries, again, depends a lot on the biological question, right? So if you're looking for kinases or if you're looking for other processes, it may be better to screen a focused library than a general diversity chemical library. And of course, the number of compounds varies depending on the types of libraries. Pharma companies will have libraries in the range of millions of compounds. Academic labs will screen hundreds of thousands of compounds typically. If we consider genomic libraries, again, here we have different choices. We can go for single oligonucleotide libraries for siRNAs and CRISPR, for example. We can do also pools. We'll come back to this in a little bit. And again, like before for the chemical libraries, we could screen for the whole genome if we have a broad question, or we can go for more targeted screenings if we have more specific questions. So for example, again, if we come back to the example of kinases, if you want to find a kinase that does a specific phosphorylation, we could just screen for the kinase library or for the phosphatase or protease or ubiquitin-related genes, and so on and so forth. So really, we have a very comprehensive toolbox of molecules that we can use for these rate screening, and these range from RNAi, microRNA type of library, CRISPR libraries, gapmer libraries, small compounds, and also cDNA expression libraries. Okay, so what I'll do now is just go through these different types of libraries and highlight some key points that are relevant when we consider screening with these different molecules. And so starting with RNA interference, as you know, Nobel Prize 2006, the discovery of this mechanism of interference of messenger RNA by RNA, determined by the sequence of the RNA. So I don't want to explain you how sRNAs work. You know that. What I want to highlight here is different ways in different systems to induce RNA silencing. So if we start here, this would be the way that we use for screening, using synthetic sRNAs transfected into the cells. This gets incorporated into the RISC complex, RNA-induced silencing complex. Again, our messenger RNA is cleaved. You cleave the message, you don't have protein. This is how sRNAs work, right? You see here easy RNAs. Easy RNAs are enzymatically produced sRNAs. And these are, you can consider them as very high complexity pools. Okay, so they are, because they are produced enzymatically from a big double-stranded RNA, you'll get multiple sequences that will cover the gene of interest. So this can also be produced enzymatically, but in vitro and then transfected to the cells. We have here this possibility as well to introduce long double-stranded RNA into the cells to get the kind of same effect as the easy RNAs. This is only possible in Drosophila cells, not in human cells, because in human cells, this would trigger a very strong interferon response. In Drosophila cells, you can do just feed the cells with double-stranded RNAs. The cell will chop these RNA into small RNAs that then will do the interference. And then we also have the possibility to encode the sRNA in a plasmid or in a viral vector in the form of a short hairpin that then is processed, is exported to the cytoplasm and processed and then incorporated into the RISC complex. Okay, so a couple of points that I wanted to make here that are important is the risk, right? So the risk exists in the cells, not for us to do gene silencing, but because it's part of the microRNA machinery, right? So basically what we're treating the cell by feeding the cell with an RNA that gets incorporated into this complex. And because we designed this RNA to be fully complementary to the target sequence, we have cleavage, okay? This is very important. Only when you have full complementarity between the sRNA and the target, you have cleavage. And this will be important when we discuss microRNAs. So what should we consider and what is the consequence of all these to screening? So we have to, we'll always have false negatives and false positives when we're using sRNAs and these types of methodologies. False negatives here come mostly from the fact that our organisms are highly redundant. We're silencing one gene at a time, or knocking out one gene at a time. So if we have two genes that perform the same function, if we knock down one, the other one will cover and the same the other way. So you will have false negatives, not because the proteins are not important, but because there is functional redundancy. The other source of false negatives is the inefficient silencing. So it may be that the residual levels of the gene are sufficient to allow for some protein production that is sufficient. And so the consequence of this is that not all factors that are involved in any given process will be identified. Okay? Then we have also the other side of the coin that are the false positives. And here, false positives are mostly due to the fact that RNA interference hijacks the microRNA machinery, right? And so an sRNA has been designed to be fully complementary to the target to induce the cleavage. But while doing so, it will also be partially complementary to other sequences. And so it has the potential to work as a microRNA and so lead to repression of genes that are not intended targets. And this is what we call the off-target effects of sRNAs. And the consequence of this is, of course, that we're going to identify some genes that are just false positives and they are not really involved in whatever process we are studying. Okay? So to try to overcome these limitations, there are also other things that one can do, and these libraries that we use for silencing also incorporate some of these modalities. So you may have asked yourselves why, when you do an experiment, you buy an sRNA pool and not a single sRNA. And this is related to the off-target effects. So the concept is that the off-targets are, of course, concentration dependent. So the more you have the RNA, the more likely it is to bind to the specific sequence, but also to produce an off-target. So if you design a pool of sRNAs targeting the same gene, the same coding sequence, you are keeping the amount of sequences that are on target, but because each of these sequences will be diluted, the off-targets will be diluted. So you are keeping the on-target, decreasing the off-target, right? And this can be taken to an extreme with the easy RNAs, where we have such a complex pool that every sequence is different from the other. So we have, instead of 4 different sequences, we'll have 100 different sequences targeting the same gene. And here you can also dilute this. So you can also do other things, that is modify chemically the sRNAs to favor incorporation into the risk of the desired strand. And you can also combine these. So for example, our libraries for screening with sRNAs are composed of 4 sRNAs modified so that they are best specificity possible. So this is the strategy to minimize the off-targets. We still have off-targets, and then typically what we do is that we screen with the pool, and then in a validation stage, we deconvolute and we test each individual sequence. And the idea is that many or the majority of the individual sequences should recapitulate the effect of the pool. If not, it means that the pool is being dominated by an off-target effect, and what we're observing is indeed an off-target. Okay, so moving to the way that we introduce these sRNAs into the cell. This is the molecules that we use for mammalian cells. We transfect these into the cells by using lipid transfection. As I mentioned before, we can, in Drosophila cells, use the long double-stranded RNAs. They are taken up by the cell and processed, or alternatively, we can also process these long double-stranded RNAs outside and then use the resulting easy RNAs to transfect the cells. For pool screenings, because these are encoded by plasmids or virus, we use short AirPs that then are processed inside the cells and then give rise to the short interfering RNA. Okay, so this is critical points about sRNAs. So microRNAs regulators, post-transcription regulators of gene expression. So this was a Nobel Prize last year. So these molecules are small RNAs, 20 to 20 nucleotides. We have about 2,500 encoded by the human genome. The very important part of the microRNAs is that they silence the expression of targets, but they do this by partial complementarity with the target. Not like the sRNAs that are synthetic, that are designed to be fully complementary. The microRNAs have evolved to be partially complementary, and this leads to modulation of the gene expression, inhibition of gene expression, not by slicing of the RNA, but by other mechanisms that involve translation repression and RNA degradation. Very interesting and very important is that each microRNA can regulate multiple targets. Okay, so because the sequence, the recognition sequence is smaller, of course, this smaller sequence is the likelihood of finding the same sequence throughout the genome is higher, and that's why this happens that microRNAs are more promiscuous, if you want, in the sense that they have naturally more targets. And what also is interesting is that the other is also true. So each transcript can be regulated by multiple transcripts. So this generates really very strong and intricate networks of gene regulation, and more than 2/3 of the genome is predicted to be regulated by these 2,500 microRNAs. So they are important for everything, basically, that have been characterized in multiple physiological and pathological processes. The recognition of the microRNA with the target transcript is mainly guided by the seed sequence of these first nucleotides on the 5' of the microRNA. And then there are some compensatory binding as well, with typical formation of these bulges. So this is the partial complementarity that I was referring to before. One very important thing you will see in some or many textbooks and papers and so on, a cartoon like this, where the microRNA is binding on the 3' UTR of genes. Yes, that's true. That's the main site where microRNAs bind. But microRNAs can also bind in the coding sequence, can also bind in the 5'. Okay, so although they are mainly targeting 3' UTRs, there are also other regions that can be targeted by microRNAs. So this is important when we are then trying to discover the mechanism of action of microRNAs, relevant targets, and so on. Okay, just to reinforce here this network of regulation where one microRNA can regulate multiple transcripts, each transcript regulated multiple microRNAs, which is great because as a screening tool, it's fantastic because we introduced one microRNA in the cell and we know that we are changing multiple genes. And when we have very complex phenotypes where we are changing the program of the cell and so on, it's very likely that changing one gene is not enough. But with the microRNA, we can trigger these changes. It also is a big challenge then when we try to characterize what are the targets of the microRNAs. And from the targets, which ones are relevant. So if we do a transcriptomic analysis of cells expressing one specific, overexpressing one specific microRNA, we typically find hundreds of genes change. So from going there to identifying which really is the critical gene is a challenge. So for screening, what tools do we have available? So this is just a different representation of the microRNA processing and action here in the messenger RNA. We have essentially two types of tools: microRNA mimics that mimic microRNA, so they get incorporated into the RISC and they exert their action as a normal mature microRNA. And we have microRNA inhibitors. These microRNA inhibitors are sponging the microRNAs. So basically, they are single-stranded oligonucleotides that bind with very high affinity to the mature microRNA. And so the microRNA is busy binding to these oligos and is not able to bind to the target.
In that example, is it a perfect match?
In that example, it's still with the bulge so that there is no cleavage result. And of course, there are genome-wide libraries for mimics and inhibitors so that we can do the screens. Okay, this was about microRNAs. If we move now to CRISPR for genome editing, again, Nobel Prize a few years ago for not for the discovery of CRISPR, but for the adaptation of the system for genome editing. This system that evolved in bacteria as a protection mechanism against invading DNA that incorporates pieces of the DNA into the genome of the cell to then produce an RNA that then recruits a protein to cleave DNA. So this can be adapted now to do engineering. So what we need is an RNA that will target the desired sequence. We need the enzyme. And so when the RNA, this complex of the protein with the RNA finds the target sequence with the vicinity of a PAN signal, then there is the cleavage of this double-stranded DNA. So this is the mechanism of CRISPR for cutting DNA. And this has been adapted to introduce mutations and to generate knockouts, because after the cutting by CRISPR, the cell tries to repair this double-stranded break. By repairing this, because this non-homologous and joining mechanism is much more prevalent but is error-prone, this will typically lead to insertions, deletions, and you get a missense mutation and the effect of knockout. This is the typical or the initial application of CRISPR that can be used for screening in a pooled format with libraries of guides, and also in an array format with synthetic guides. Now, even more interesting than the CRISPR itself that cuts is the adaptations that were made afterwards, generating a dead Cas9. So by mutating these two nucleolytic sites in the enzyme, this dead enzyme was created. And the good thing about this enzyme is that it retains the ability to target the DNA sequence by providing the RNA, but does not cut. And this has been a major advantage, because now we can use this dead Cas9 as a platform to tether other proteins and to bring these proteins to specific genomic locations in the genome. So you can see here just some examples. So this is the typical example of a normal CRISPR that cuts, but then we can fuse the Cas9 with a repressor or an activator to get gene regulation, epigenomic modifiers, fluorescent proteins, dimers to create looping and change the topology of the chromatin, target RNA as well, base editors. So different types of approaches that we can use now with this dead Cas9. And the most interesting, I think, in the context of screening and regulating gene function are, of course, transcriptional regulation, right? So now we can use this dead Cas9, fuse it with transcriptional repressors, and repress the transcription of specific genes. Or even better, doing transcriptional activation. And I say even better because there's not many tools that we can use to increase expression of a gene, right? So I've shown you microRNAs, siRNAs, CRISPR knockouts. These can all be used to loss of function. But for gain of function, we're basically limited to cDNA overexpression, right? With virus or not. And so here opens the possibility now to do genome-wide transcriptional activation mediated by CRISPR. And this opens new opportunities for investigating gene function. Okay, so I think this is really exciting for screening, because now we can do both things, activation and inactivation. So this process here of CRISPR, of repression by CRISPR, it's called CRISPR-I for CRISPR inhibition, and this one is called CRISPR activation, so CRISPR-A. So if you see CRISPR-A, CRISPR-I means using of these modified Cas9 enzymes to modulate transcription.
Can I ask a question? So switching these genes off and on with Cas9 is fantastic, but I'm just wondering what happens if you want to activate a gene where the chromatin is very inaccessible. So is this still a limitation?
It is a limitation, right? And because of course, either for that you would have to modify also the chromatin context and so on. So of course, it will only work for certain genes, right? So I don't have experience enough with CRISPR to tell you what is typically the efficiency of upregulation that we can get and the percentage of genes that will respond. But of course, chromatin context will be an issue for many of the genes. Yeah. Whereas, for example, if you think of the other systems, RNA interference and so on, the RNA is already produced, so it's not an issue. So yeah, if the genome locus is really, it can be that it will not respond just to a simple activation. Yeah. But still, for many genes it works, and it's a fantastic tool with all the limitations.
Can I ask what size Cas9 is in its I or A form?
Yeah, I don't have the numbers out of my head. They are not huge in the sense that they can be cloned. I'm not sure if in AAV, but they should be a few kbs long. So it can be transduced even as a messenger RNA into the cells. For example, one very easy way to achieve this activation is to transfect cells with a messenger RNA for this activator or inhibitor together with the guide. Right? Because remember that for the sRNAs and microRNAs, the cell has all the machinery already.
Yes.
The risk. But for CRISPR, you have to introduce the enzyme, whether it's the native Cas9 or the Cas9 for activation or inhibition. But it's not big, so that it cannot be transferred as a messenger RNA or as a plasmid or a vector.
Cool. Thank you.
Okay, good. And just to finish, also a quick word about long non-coding RNAs. So we know that only 2% of the genome, our genome, human genome, encodes for protein coding genes. But the truth is that most of the genome is transcribed. 80% of our genomes is continuously being transcribed. And a lot of these RNAs are what is called long non-coding RNAs. They are called long just to distinguish them from the short RNAs that have been arbitrarily considered above 200 nucleotides. We don't know how many long non-coding RNAs we have in our genome. There is estimates from 20,000 to 150,000, so this means that we have no idea. There's a lot of long non-coding RNAs. They don't have specific features nor specific mechanisms of action, so that's why it's so difficult to understand their function, their mechanisms, and their number as well, because they have different mechanisms of action. So they can, act as scaffolds. They can regulate regions in the genome. They organize protein complexes. So very diverse functions. It's not like the microRNAs that we know the mechanism of action or other types of molecules. So this is really a diverse, and they're very specific to the cell type, right? So tissue and developmental very tightly controlled. So different cells have different pools of long non-coding RNAs depending on their status and origin and so on. Also, they are nuclear and cytoplasmic. Some act in the nucleus, some act in the cytoplasm. So it's really difficult to work with long non-coding RNAs, and even more to screen microRNAs, because this is very context dependent, right? So there is no genome-wide library of molecules to target long non-coding RNAs. There have been attempts to do libraries, but these libraries are like the 2,000 most conserved long non-coding RNAs found in most cells. These are the libraries. So typically what people do that work with long non-coding RNAs is that in their specific system, they sequence what are the links that are there, and then eventually they target these long non-coding RNAs that are differentially expressed or of interest. And to target long non-coding RNAs, the tool of a choice, and this is why I brought this slide, is what is called the gapmer. Okay, so these gapmers are molecules that are synthetic molecules. They are hybrids of RNA and DNA, LNA is this modified RNA that is much more stable. And so the way that this works is that these molecules are designed to be complementary to the RNA. But because they have this DNA portion here in the middle, when they bind to an RNA, they create this heteroduplex, RNA-DNA heteroduplex. And this heteroduplex is recognized by the enzyme RNase H that we have in our cells. So it's a mechanism that is a mechanism of RNA degradation, artificial RNA degradation triggered by this enzyme. One advantage, or the main advantage of this, is that this also works for nuclear long-coding RNAs. Because these RNAs are in the cytoplasm, the risk machinery is in the cytoplasm, so it's difficult to target nuclear long-coding RNAs. Whereas with this technology, because the enzyme is also present in the nucleus, we can also target these long-coding RNAs in the nucleus by transfecting these molecules. Of course, these molecules have to be designed for the specific RNA of interest. Okay, so this is about the different systems that we have to modulate the gene expression. I did not mention base editing, which is a variation of Cas9, where you have then an enzyme that will do editing of the base that can also be targeted for screens. Okay, so before we move on to other part, let me just emphasize here one point, that is doing knockdowns by RNA interference or by CRISPR-I is not the same as doing knockout with CRISPR. So if you think of cells treated with siRNAs or cell treated with CRISPR, this would be, you are seeing different things when you analyze these cell populations. So when you do, for example, CRISPR knockout, the outcomes of this knockout can only be these three, right? So either you have the cells that have not been edited, or the cells that have been edited in one allele, or the cells that have been edited in both alleles. Okay, let's consider the diploid cells just to keep our lives easy, right? So you will have a population of cells that will respond in one way, and other in another way, and the wild type cells. When you're doing an siRNA treatment or a CRISPR-I treatment, what you're doing is you're affecting the levels of the RNA in all cells. So you're shifting the population towards lower levels of RNA. So it's to be expected that the response to a knockout or knockdown is different. Okay, and this is of course even particularly more critical for essential genes and genes that cannot be completely deleted but can be knocked out. So you can analyze these, but not that phenotype. So this is just a conceptual difference that we should have in mind when we do these types of experiments, not only for screening but also normal experiments. So to wrap up this part, just a few more points. That is, what are the requirements and challenges of doing these large-scale experiments? So as a requirement, we need to have a very robust and reproducible assay because we're testing multiple treatments. So these have to be very consistent so that we can analyze differences, and it should be as simple as possible. Okay, we should not overcomplicate our screening. We should do enough to answer our biological question, but not overcomplicate the screening, because this then may compromise the rest. As for any other experiment, we need very good positive and negative controls. We need an assay that is scalable, cannot be an assay that can only be run in a 6-well, right? Because we cannot do screenings in 6-well. And if we're considering genomic screenings, the cells has to be transfectable with synthetic molecules. In terms of challenges, this is slightly you can read it. Okay, so because these are cell-based assays, these are always less reproducible than biochemical assays when you have just, a recombinant protein and a drug, for example. The protocols need to be optimized for every cell line, for every readout. So it's not that because we have done one screen, we can use the same assay or for other cells or for other types of readouts. We have to, of course, there are some things that will be the same, but we need to optimize every time. If you consider microscopy screenings, we have to also consider the large data sets that will be generated. Off targets, we spoke about off target effects. CRISPR screenings, we also have to think about kinetics, penetrance of the phenotype, and the fact that we need to introduce the enzymes inside the cell, which brings an additional level of variability. And then, of course, we're doing large-scale experiments, so we'll end up with large numbers of targets to validate and to follow up, which is also a challenge. Okay, so very briefly, to give you an idea of what is the kind of infrastructure that we need to run screenings, we need dedicated equipment, like the microscopies, content screening microscope, liquid handling platforms for handling all the libraries and so on, dispensers, washers, and then depending on the types of screening that you are doing, you may need, long-term sterility if you're using live cells, or, for example, nano-dispensing capabilities if you're doing, for example, drug screening where you want to dilute your MSL vehicle for screening. These are just some examples of microscopes. They have different types of illumination. These days, they are all equipped with confocal spinning discs, environmental control so that we can do kinetics, CO2 and temperature control, transmitted light, and multiple illumination sources, multiple filters, so we can multiplex. You've seen examples with 5 colors, 5 channels that can be done. For the liquid handling, we have the traditional liquid handling machines using kits that pipette in the microliter range. But there are also other systems. We don't have these at King's, but we have these other systems that are contact-free system based on acoustic dispensing, and these have capacity to pipette in the nanoliter range. So directly from stocks, you can, by using ultrasounds, project droplets of the liquid to the destination wells to do nanoliter volumes.
Can I ask you about the one that we have? Have you ever worked with any cell types that are disturbed through liquid handling? So do you lose cells from your wells after fixation, washing, staining, washing, imaging?
The answer is yes. We have this, so any cell will be disturbed if you don't wash it properly and if you don't fine-tune the parameters properly.
Mm-hmm.
There are some cells that are more sensitive than others. For example, I can tell you that you cannot wash 293s with an automatic washer because they fly away, right?
Right, okay.
Because they are so poorly attached, unless you use coated plates or some tricks. But for a lot of other cells, we managed to find the sweet spot so that we can wash them and not detach them with the automatic handling. This is done with plate washers. The washing is done with these machines that what they do is that they aspirate the content of the well and then they dispense the washing solution. You can define different parameters, speed, height, and so on. What we typically do is that we leave some volume at the end of the well so that it cushions the liquid that is coming in. And by doing this, you can get a good compromise between detachment and washing. There are some problematic cells, but I mean, there's always, or usually, a good alternative. We also have other types of machines, like Washer was saying. For example, the reagent dispenser for plating cells into the plates, or the sealers to seal the plates. And I skipped this one, that are the plate readers, that is the alternative readout. So when we don't use microscopy, we can also use reader for, more conventional technologies, absorbance, fluorescence, luminescent, but also for more advanced, like alpha screen, time resolved fluorescence, or polarization. Okay, so this is our infrastructure at King's. You have seen it. Some of you have worked there. So this is a fully automated platform where we have the high content screening microscope and some equipment. And if I show you now the components of the system seen from the back, so what you see here is the microscope that does the automated reading of the plates. We have two liquid handling machines, one with a 96 head for reformatting plates, one with 8 independent channels to cherry pick compounds or molecules from source plates and do custom libraries. We have an incubator on deck when we need to do, for example, kinetic measurements and keep the cells in the incubator. This is fully automated. We have a plate sealer, a plate reader, an hotel to keep tips, plates, and so on. And then we have this robotic arm here that is on a rail so that it can access all the instrumentation and bring the plates to wherever they are needed. In terms of library, our focus is on functional genomics. So we have libraries of sRNAs for mouse and for human. We have the pooled version of the libraries for independent sRNAs, per gene inside each well. For the microRNAs, also have different libraries, mimics and inhibitors. We have some libraries of small compounds. These are mostly FDA, EMA approved compounds that we use for pathway discovery or for repurposing purposes. We have also a library of the mouse secretomes. So this is encoded in viral vectors that we can also use for screening. And then we have the ability to do custom libraries from any of these libraries, but especially from the sRNA library. So if we are interested in a specific protein class, kinases, phosphatases, proteases, we can just screen that subset. Or even more interesting, if we have a gene set of interest that is derived from RNA-seq, single-cell-seq, microRNA, target identification, whatever, we can just cherry pick these genes of interest and screen only those. So this is what we can do. And of course, if there is interest in other libraries, we can also buy these libraries and screen them for specific purposes.
Much library.
That's a difficult question because it depends a lot on the amount and the complexity of the number of genes and so on. So for example, I can tell you that we're looking now to buy a library of covalent compounds. 3,000 compounds cost 10,000 pounds. The libraries of genome-wide sRNA libraries range in the 100K range, the genome-wide. So ballpark, they're expensive, but also they allow us to do multiple screenings. Okay, so it's not that we buy a library do one screening and the library is on, right? So we can keep the libraries, and we have some of these libraries for years, and we can use them until we run out of them. Then also depends on the cherry picking, right? Because we have genome-wide library. Every time we do a cherry picking, it's a screening that you can no longer do because that gene's missing, right? So it's always a compromise between, but we still have enough of the libraries to do all these things for now. Okay, so quick few words about assay development. This is really the most important part of the screening, where you need to consider all the parameters to get your assay ready to screen. Again, as I said before, there's different options for screening, and we should consider what type of information do you need and decide what type of readout do you need. And then this, we don't need to overcomplicate the screening. So we should keep it simple and answer to the biological question, and then do further studies afterwards. So you can go from simple biochemical assays if your target is one specific enzyme, or you can go for cell-based assays or high content or live cell or profiling, depending on this, knowing that there is always a trade-off. Increase complexity, you decrease throughput, you increase cost, and so on. The same for the types of cells that we're using as models, right? So engineered cell lines or cell lines compared to patient-derived cells or iPS compared to even more sophisticated systems, there's always a compromise, and this needs to be really well balanced. And the other thing is also miniaturization. So it's different screening on a 96 or a 384 or a 1536. Well, so we don't have capabilities to work with 1536 here, because you need very precise equipment for pipetting and so on. But 96 for a genome-wide is really a lot, right? Because we're talking about 200 plates. So all the screenings that we do are on these 3 to 4 well formats. We can go to 96 for specific purposes, small screens if needed, but in our experience, almost everything works in 384. We have a lot of experience there, and this is all manageable in terms of volumes, evaporation, and so on, and throughput as well. So I think it's 480 has been the best compromise. So as a general workflow for screening, this is the initial part where we go from the assay, bring it to a status that can be screened. Then we do the primary screening, analyze the data, identify leads, and then either we go for follow-up or we do secondary rounds of screening, depends on the screening. So the job in the lab is that we spend 3 months doing assay development. We spend 3 weeks doing the screening, and you aim at spending 3 years doing the follow-up. Okay, so this is for the ones that are starting the PhD to have an idea of how it goes. So what do we do during assay development? All this and probably more, right? So we have to consider all these things about your assay, cells, transfections, availability, banking of the cells, controls, transfection protocol, forward, reverse, reagents, amounts, concentrations, and then the assay itself, antibodies, stainings, and so on, analysis as well, automation, washing, very critical, because this is very cell type dependent as well, and readout, right? The aim is to get this as reproducible as possible, from well to well, from plate to plate, from day to day, and if possible, even if it's a different person doing this But typically, it's not the case. But we need this reproducibility because, especially for the large-scale screenings, these are not run in one batch. So these are run in multiple batch across different weeks. So if the batch from the first week is very different from the batch from the second week, then we cannot compare, and then this will affect the outcome. So we really need reproducibility here. And then controls, right? So in all plates, we put different types of controls. Positive controls that give us the phenotype that we want. Negative controls, and that should be as neutral as possible. And we also use, in the case of when we do transfaction controls to make sure that if we don't see an effect, it's not because the transfaction didn't work. It's because there's no effect, right? Because there's always this problem that is, if you don't see an effect, is it because transfaction didn't work, or is other reason? And so because we keep these controls in all plates, we can do immediate quality controls of each plate, and if the controls in the plate do not behave as they should, this plate can be discarded, and this way we get more and more confidence in the results if there is a technical problem happening. How do we measure how good is the assay? Well, if you do enough screenings, then you get a feeling of how good is an assay just by looking at the data, and it's really easy, right? It's like any other experiment. What you want is very good separation between your positive and negative controls, and narrow distributions within each treatment. So this can be measured. There is this Z prime factor that can be measured for any specific readout. This is mostly used for plate reader-based assays, because in high content, in microscopy, it may be that one single parameter is not good enough, but it's the combination of multi-parameters that give you the robustness of the assay. But still, you can do this type of metrics to understand how good is your readout. And the rule of thumb is that if it's greater than 0.5, it's great. And if it's less than 0.5, if it's a biochemical assay, you don't even try it, you don't even screen. If it's a cell-based assay, it's up to you. We have screened with much less than these, and with microscopy, we can still get very good data. I have a quiz for you now. That is, we have here 3 examples of positive and negative controls, right? A, B, and C, the same assay. Of course, no one doubts that this one is the best, right? Big separation, narrow distributions. So my question is, between this one and this one, which is the best?
I would say C, because maybe you could tweak it to separate them out a bit more, but the variability
Could be.
hopefully will stay the same.
Exactly, could be, but what you're going for. This is probably better. It's probably easier to get a narrow distribution here than a bigger separation there, because there's this variability. Probably you can work on it to make it less variable with automation, with tweaking some parameters. But this is to tell you that, I mean, these 2 components are very important. It's not only how different is your positive and your negative, it's also how variable they are to really make the assay a good assay. Okay, so I can go quickly through this part of the models. I'm almost finished. So just want to touch on these models and then give you really 2 slides each, 3 examples of screenings, just to wrap this up in the context of cardiovascular. So about the models, there's mainly 2 or 3 trends when we think of more physiological relevant models. One is 3-dimensionality, getting from 2D to 3D, because we are not 2-dimensional. The other one is going for iPS-derived cells, patient-derived. The other one is CRISPR-edited cells. So I think that it's clear what are the advantages conceptually of working 3D versus 2D, but there's also some problems, right? So the challenges of working in 3D is this reproducibility that we're speaking about. In 2D, sometimes is already challenging. If we now imagine 3D and you imagining scalability of these 3D structures, if you imagine imaging of 3D structures, if you imagine transfection of 3D structure, this all becomes much more complicated. It's possible, depends really on the system, but it gets much more challenging to do, especially large-scale screening in 3D. So depending on the question, this can be done. For drugs, it's easier because you remove the problem of the transfection, but for doing genomics in organoids is complicated. So yes, these advantages of the 2D become more determinant. And so most of the screenings are done in 2D and then validated in 3D. iPS-derived models. So of course, this is if we can do this would be super interesting for screening. We mostly use either cell lines or primary cells from animals. If we can start using iPS-derived cells, cardiomyocytes, fibroblasts, endothelial cells, I think this would give us a very strong advantage. There are, for some of the cells, we can use primary cells. I'll show you an example later with primary fibroblasts that we did. It's possible to screen with the iPS-derived cells. The problem is, again, there are some challenges, right? So the advantages, I think they are very straightforward. The challenges is mostly due to the time of the differentiation. And now we transfect these cells or we treat these cells along the differentiation protocols with change of media and so on in multi-well-plex, right? One limitation. The other one is the availability of the differentiation protocols and the efficiency of these differentiation protocols. The other ones, or this one is more conceptual, so how far do these cells recapitulate the adult phenotypes? But I mean, again, most of the problems are on this scalability. I don't think the scalability here, the issue is the number of cells, because we know that we can get high numbers of cells, is really if we can get these cells to be usable during the time frame of the assay without medium changes and so on. So this is another point. And the other one that could also be plugged in, the other ones, is to use CRISPR cells to create isogenic models, for example, or to validate targets or to create cells for then doing screening. So you can use CRISPR in different ways. Here, again, advantages and disadvantages, and the main advantages here would be that we can combine, and if we want a really sophisticated model, combine CRISPR with iPS, create an isogenic model, and then compare treatments and so on, bringing all the advantages that we have seen from the iPS. Okay, so typically our process has been to screen in simpler models and then do the validation in these more sophisticated models. But more and more we want to screen with these especially the iPS-derived cells, so that we can do it in the human context and not mouse, or at least particularly for the cardiomyocytes. But again, this is a trade-off again, and depends on the phenotype that we are interested in looking. Okay, so just to finish up, I'm just going to show you 3 examples really quickly, not going into the science, but really on the screening of 3 examples with microRNAs. And in this example, what we did was to measure the size of the myocytes, looking at microRNAs that would increase or decrease the size of the cells. So very simple screening, reverse transfection of the microRNAs into rat cardiomyocytes, waiting 72 hours. We also measured proliferation, it's not here. And then fix the cells, wash the cells, stain the cells, and then look at the size here. And you see here the original images, you see the computer-generated images, and then you can, for each cell, not measure thousands of parameters. In this case, we measured area of the cell and then score them as pro-hypertrophic or anti-hypertrophic, just to look at how this could then be used. And then as a secondary assay for this, looking at more specific markers, in this case, ANP, that localizes in this perinuclear localization when there is this pro-hypertrophic stimuli. And then you can see here a protocol with these microRNA cells getting an ANP and bigger. And here the reverse protocol where we stimulate the cells to get hypertrophic and then counteract it with the microRNA. And you can see a reversion of this phenotype by these microRNA. So very simple screening. And then this was done as a secondary screening, just for the ones that have scored in the primary screening. This is one example. Another example, in collaboration with Andy Baker at Edinburgh, what we look at here was microRNAs controlling endothelial to mesenchymal transition. Here we use UVEC cells stimulated to undergo EMP. microRNAs, this screening by microscopy again, using 2 specific markers, one of endothelial cells, the other one of mesenchymal cells. And what you can see here is that unstimulated cells, you see very strong CD31 staining. When you stimulate the cells, we get this Langerhans staining, so very strong, whole cells red. And then these cells stimulated with microRNAs that we identified as top of the screenings, they revert completely to a non-stimulated phenotype. So these are potent blockers of these processes, and we are following up on these now.
So is this like, sorry, I missed your workflow.
So we stimulate the cells.
But you reverse transfect at the beginning.
Yes.
Rather than add it afterwards to revert the phenotype.
No. So yeah, you're right. So we are preventing the phenotype from developing, if you want. Now, these are the follow-up assay. So the way that we designed the screening was like this, to do the reverse transfection because it's easier from the automation point of view.
Yeah.
And so what we did was, it's true that this is a setup in which we are preventing. And now for the topics, what they have done is to do the other scenario, that is the reversion phenotype, where first let the phenotype establish, then you transfect to the primary. This is a second step, because it would increase a lot the complexity of the screening, the foreign transfection. So we went for this option. Okay. And one final example on the microRNA is for modulation of cardiac fibrosis. And here what we did was to use actually human primary cardiac fibroblasts and look at different phenotypes, proliferation, activation, and collagen deposition. And again, we find different microRNAs modulating these different phenotypes that we are now investigating. So this was just to give you an example of the 3 different cell types, art-related screens. Of course, we have done many others, not only in the cardiovascular system, but also there. And with that, I'm done. I hope I show you how powerful this technology is to identify novel targets, to develop novel therapies, and mainly to discover new molecules and new biology. Thank you very much. If you have more questions, and people online as well, if you have any questions, feel free.